So this is going to go with chapter 14 in your book, and we're going to start it by talking about what is the central dogma of molecular biology. Now, the central dogma of molecular biology is the hierarchy of DNA flow in biology, which is to say, how does DNA, RNA, and protein interact? What's the pathway? What's the flow of information? Now, you may have had some of this in your introductory biology labs, and some of you may even have a question that deals with this. So let's talk about it. Okay, we start the central dogma with DNA, deoxyribonucleic acid. DNA will code for RNA. RNA then serves as the basis to make protein. All right? Now, going from DNA to RNA, this is transcription. Okay? Just like you transcribe something, for example, taking one thing and writing it down and making an exact copy of it, that would be transcribing something. Well, here, transcription is where we take the DNA and make a short copy of it called RNA. Now, RNA going to protein, this is translation. Think about a translator taking one language to another language. Okay, that is translation. Now, this is my analogy for it. Now, think of your genome as a hard drive on a computer. Okay, that hard drive has thousands and thousands and thousands of files in it. Okay, our genome has thousands and tens of thousands of genes in it. Okay, now let's go back to that hard drive analogy. Let's say I have a file and I want to share that file with someone. Okay, you have a file, you have some test notes that you want to share with me or I want to share with you. Am I going to take my entire hard drive out of my computer and say, here's my hard drive, it's all yours? No, of course not. Why is that a bad idea? Because you don't need all of those files. You need one file out of thousands. And also that hard drive represents your original files. Okay? So it's not a great idea to share all of those files at one time. Well, similarly, when we want to share a gene in our genome, we don't open up our entire genome and say, hey, we need one of these genes, so we're going to expose the entire genome. No. So what do? Well, let's go back to that analogy I'm using. If I want to share a file with you, I'm going to share it on a thumb drive, right? I'm going to take that file, I'm going to put it on a thumb drive, and I'm going to say, here you go, here's my file. All right? Well, in the parlances of biology, we're going to take RNA and make a copy of a single file. Or in this case, we're going to take RNA and make a copy of a single gene. Sometimes in bacteria, we can actually make multiple genes on a copy. But for the human example, we're just going to talk about one gene on one mRNA. All right? Then I'm going to go and take that thumb drive and put it in a computer, and then I'm going to print off something. And what I print off will be the proteins in this analogy. Now, what's a benefit of this? What is another inherent thing here? Well, first, obviously, is a protection idea, right? Our original file on our hard drive is protected because we're only sharing copies of it. Okay, so we're not sharing all of our files at one time. We're just sharing a copy of it. Okay, so RNA is just a copy of a single gene. Now, what would happen if you lost that thumb drive? You go to Walmart and buy another one for $2. They're cheap. Okay, RNA in the cell, same idea. You know, RNA is an ephemeral molecule. It doesn't last very long in the cell. Okay, so something happens to that RNA copy, so what? Okay, the original is safe in the genome. Just like in this example, in this analogy, the file is safe in the hard drive because we did not share the hard drive. We just shared a copy of a file on the hard drive. All right, now going back to the idea of translation and transcription. When I have a file on my hard drive and I make a file on a thumb drive, that's not a vastly different format, is it? No, not really. Okay, when we go back and look at the central dogma of molecular biology, DNA going to RNA, these are comparable molecules. Now, there's some differences here we'll get to, but DNA and RNA are both nucleic acids. Okay, so this is transcribing something. We're not significantly changing the underlying format. These are both nucleic acids. Some differences, yes, but for the most part, they're very similar. We'll talk about that in a few minutes. All right, now think of going to protein. Protein here is analogous to a printed document. Do you see how different the ones and zeros are in that file on your thumb drive compared to an actual paper document? Okay, they're significantly different, aren't they? Well, coming back to this example, going to RNA to protein is like going from your one file on your thumb drive to a printed copy. These are significantly different in their format. Okay? Here, RNA is a nucleic acid. Here, protein is a polymer of amino acids. Vastly different structures. Okay? Now, what's another advantage to this model? Well, there's another thing, it's an amplification idea. So, for example, let's say I have a thumb drive on my computer. I have a file on my computer. What if I need to make lots of printed copies? Let me give you an example. I have this file on my computer, and I've made a nice test review, and I go to an area where there's a lot of fraternities and sororities. Okay, and I want to get that test review out to, 80, 100 people. So what do? I say, okay, you're in this sorority, I'm going to give you this thumb drive. You're in this fraternity, I'm going to give you this thumb drive. You're in this fraternity, I'm going to give you this thumb drive. You're in this sorority, I'll give you this thumb drive. Okay? Now, everyone takes that back to their houses, and then they put it in a computer, and they make 3, 4, 5 copies at each location. Since I have given 4 copies of the original file, and each of those files has made 4 or 5 copies, I've done what? I've made 20, 30, 40 copies. Okay? Now coming back to our model here, we have DNA, right? Well, what if we make several RNAs of the same gene? So there's one, here's the entirety of the genome. Okay? Here is the particular gene I need to make a copy of. So I make multiple copies of that gene. Okay? Now, each of these copies, these RNA copies, will serve as the basis for making multiple proteins. Okay? The pink here is meaning protein. So I went from one gene in DNA to 4 copies in RNA. And one of these RNA copies made this many proteins. And this RNA copy made this many proteins. And this RNA copy made this many proteins. Do you see the amplification going on here? We go from one gene in our DNA to several RNAs, and several RNAs will lead to even more proteins. An amplification is going on. And this model amplifies the signal. We go from one gene to several RNAs to many, many, many proteins. Okay? Just like in that analogy I gave before, we have one file. I give everyone a copy of that file on a thumb drive, and then they go back to their sorority or fraternity houses, and they print off multiple copies for everyone there. I've amplified the file. And that's what the central dogma does, is it has an amplification quality to it. All right. Now, last point here is very important. The function of a protein has been coded for by the DNA sequence. If you think about the central dogma, the protein is ultimately determined by what the DNA is. Okay, so what the protein is a function of what the DNA sequence is. Okay, now, we talked about this last time. DNA, has those four familiar bases, A, T, G, and C. Now, we had this question, and this was a huge question in the century, or last century, I should say, was how does DNA have that diversity? DNA has four bases. So someone in the 1940s looking at it might say, well, you have four bases there. How do you go from four bases, okay? All right, so how do we do this? We have four bases. How do we go from four bases to life? The diversity of life, all from four bases. How do we get there? Okay, well, the answer is not so simple as four bases. Rather, we have a sequence, a triplet we call a codon. Okay, so let me kind of explain how a codon works here. A codon is a three-base sequence. Okay, so at base 1, base 2, and base 3, or three-position sequence, I should say. And every position can be A, T, G, or C. A, T, G, or C, or A, T, G, or C. And we do all the possible permutations here, and all the possible combinations here. And what do we get? We have four possible ones here, so we put a 4 here. And we have four possible ones here, so we have a 4 here. And then we have four possible combinations here, so we put a 4 there. Okay? These are three independent events. We do the math on this. 4 times 4 times 4 is 64. So if I was to sit here and make all the possible combinations of A, T, G, and C for these three positions, I would see that I had 64 possible combinations. So instead of us having an alphabet of 4 bases, really we have an alphabet of 64 codons. And as we'll see pretty soon, the codon is the basis for one amino acid. So a codon codes for a single amino acid. And we'll talk about that coming up shortly. Okay, so transcription. Transcription is the idea of going from RNA to DNA. Transcription. Again, think of when you transcribe something. You're writing it down. You're using basically the same format, right? Say I say, here's my list from Walmart. Transcribe this list I want you to get from Walmart. What do? You take my list on a piece of paper and you write next to it. He wants some sodas, he wants some cups, he wants this, this, and that. Okay? That's like transcribing something. Now here we're using the same basic format. We're going from one type of nucleic acid, that being DNA, to another type of nucleic acid, and that is RNA. Okay? Now let's talk about RNA-DNA differences. Now they're both nucleic acids. Okay? But RNA is going to use uracil instead of thymine, and RNA uses ribose instead of deoxyribose. Now when you think of DNA, the D in DNA stands for deoxyribonucleic acid. It has a deoxyribose in it. So let's look at that structure first. Okay, now let's look at that 2' carbon. Do we see we have the guanine base and then we have that orange hexagon? Starting from the right, we have a 1, 2, 3, 4, and a 5. Now look at that 2. Do you see the oxygen hydrogen? This is ribonucleic acid. This is deoxyribonucleic acid. Do we see that we have a hydrogen instead of an oxygen hydrogen? Okay, deoxy, Latin from or away from is de, like if you're defunct, or to deforest, right? You take that away. Oxygen's been taken away, all right? Well, here, DNA has missing oxygen, whereas RNA has that oxygen. Okay, so that's the major structural difference. Now that will lead to a difference in how these function. Now, your RNA uses uracil instead of thymine. Okay, so before, we remember we had this nice little A, T, G, C idea. Well, instead of having uracil, instead of having thymine, we have uracil. Now, there's a little way that I remember this one, and it kind of might help. Think of AU as Auburn University, and think of GC as Gamecocks, the university's mascot. Okay, so Auburn University, Gamecock. All right, I don't know if that helps. I hope it does. All right, now, RNA, when we start looking at how RNA behaves, RNA is a very ephemeral molecule. RNA does not last a long time in the lab. When you work with RNA in a lab, you have to take special precautions. It's a very difficult molecule to work with. Now, we're not going to get into the reason why now, but I'll say that RNA has a reputation for being very unstable. Okay, if you go home and dream about the RNA that you extracted today, it'll probably be degraded tomorrow. DNA, on the other hand, much, much more stronger molecule, at least in a laboratory setting. Okay, now we'll talk about that probably later on, but for now, just know that RNA does not have the long-term staying power that DNA does. Also, most RNA is single-stranded. That's for mRNA. Now, we can see double-stranded areas, for example, on tRNA, but a lot of times RNA will be single-stranded, whereas DNA will be double-stranded. All right, so again, here are the differences. DNA double-stranded, RNA is shown here being single-stranded. Difference 2, we see that DNA has the thymine. Look at the bottom, we have that T, thymine. And on the bottom here, we have uracil on RNA. Now, we're not going to go into the reason why there is uracil. It actually has to do with cytosine. If you are curious about it, ask me after class or ask me at some other point, and I'll fill you in on why we have it. Okay, but it kind of goes beyond the scope of this class as to why we have this difference. But there is a good reason for it. Okay, so we have 3 main types of RNA. Messenger RNA, and that's going to contain a series of codons. That will contain a copy of our gene. We have ribosomal RNA. Okay, that's going to be associated with the ribosome. And then we have tRNA, or transfer RNA. That's going to carry that will have an amino acid on it, and we'll talk about this later. This will have an amino acid and what we call an anticodon. And this helps to translate between our RNA sequence, which is based on a series of codons, and amino acids. And again, we will talk about this later on when we get more in depth into translation. All right, now, translation. Think about what a translator does. Okay, a translation here is going from one format to an entirely different format. Now, I gave an example before. Remember we said going from a file on a thumb drive to paper was a translation step? Well, here we're going from a series of codons on the mRNA to a series of amino acids of a protein. Okay, a vastly different change in molecule type. Before we have a nucleic acid, afterwards we have a protein. Totally different thing going on here. Okay, now we'll get again more in depth into translation later on, but now just know that when we go from mRNA to protein, we are doing translation. Gene expression. Now, analogy I like to use with this, let's say, here's a question. I think you'll probably hear this question some in your introductory biology class. And the question is this: how can you have a skin cell and a skin cell and a neuron. Why are those different? Okay, well, let's look at the genome here. Is the genome, and if I took one of your skin cells, and I took one of your neurons, and I took one of your bone cells, and I took all of these, and I did a sequence on all of those. So if I took one of your bone cells, I took one of your neurons, and I took a skin cell, and I was to do a DNA extraction on this, this, and this, all from you, and I compared the sequence of these, they would be the same. These would have the same DNA sequence, the same genome. So how the heck do we have bone cells, neurons, and skin cells that are vastly different in structure, in function, in everything discernible? How do we get there from having the same genome? The answer to that question is two words, and that is gene expression. Okay, let's use this analogy. Now, let's say that instead of a genome, a lot of people use this thing called Spotify, right? Okay, so here is the entire musical library of Spotify, right? So we've got millions and millions of songs here. So from this one single repository of music, you can pick out your own playlist. Maybe you want something from Justin Bieber. Okay, here's your Justin Bieber playlist. Here's your late 90s rock playlist. Okay? Let's see, what else we got here? Maybe here's your Debbie Gibson playlist. And everyone loves Debbie Gibson, right? I guess not. All right, but think about it that way, that you have this massive repository of songs, and you can pull out different playlists, right, from that same repository of songs. Well, let's look at this from the perspective of a genome. Instead of this being a large collection of songs, let's say that now we have a large collection of genes. And this is your genome. Okay? Your skin cell may get these genes. I'm going to use them in pink. Your neuron may get the same genes here, but different genes here. It might express these genes and the same genes here. Right? Your bone cell may express these genes and these genes. Right? So the idea here is that different cells use the same genome, but they're going to express different genes. Okay? And they're going to express them at different rates. Now, is this to say that a skin cell and a neuron and a bone cell will express different genes? Well, they might express the same genes. But then again, one might express the same gene at a low rate, while the other one expresses that at a high rate. Okay, so it is how much it expresses that, how much it uses. But the point I'm trying to get here is gene expression is the idea that our cells have a massive repository of genes. But which genes they choose to use varies. Okay, just like Spotify or Pandora or whoever, they have a massive repository of music. But when you make a playlist, you're only picking and choosing particular songs in a sea of many songs. Okay, same thing with our skin cells, our bone cells, our neurons. They have a massive number, many genes to pick from. Okay. And they will pick only certain genes to express. And express means to make it into a protein. They transcribe and translate that gene into a protein. Not only that, but certain cells, they may transcribe and translate the same gene, but they may do it in a different amount. Okay? A skin cell may transcribe and translate the same gene as a neuron. but the neuron may have a very low level of expression, whereas the skin cell has a very high level of expression. And this difference in expression makes these cells different. But they all come from the same repository of genes. Okay? Half of those genes coming from your mother, half of those genes coming from your father. Okay. Now, we're going to transcription again. We're going from DNA to RNA. Now, what does this? Transcription is done by RNA polymerase. We talked about DNA polymerase last time. RNA polymerase is the same idea. Now, let's say we have this whole, and I'm doing this to draw a genome, right? And we know your genome is, 46 chromosomes, all that stuff. But I'm just using this as a stand-in for a genome. Okay? Now, we have something called RNA polymerase. RNA polymerase is going to interact with this genome to make a copy of RNA. Okay, but remember we said RNA is like a file among thousands or tens of thousands of files. So here, this is tens of thousands of genes. We only are wanting this gene right here to have a copy of it. So what are we going to do? Well, this thing about RNA polymerase, here I'm going to just draw a little circle representing RNA polymerase. Is it going to float around in here and look for a particular thing? Is it just going to randomly guess a place? The answer to that is no. No, not really. We have a sequence before our gene called a promoter. Now think of Don King, right? Don King, a boxing promoter, right? Boxing promoters, they are a promoter in general. They get something going. They start a party. They organize a fight. They get something going, don't they? Well, a promoter is going to do the same thing here. RNA polymerase will sit on that promoter, and then it will move down our DNA, and as it does, it will make an mRNA copy of that gene. So we have a sequence preceding our gene that we call a promoter. And that's what RNA polymerase, the enzyme that makes the RNA here, binds to, connects with, and makes our RNA. Now that's a very basic description. It does get a lot more complex than what I've led on here. But for now, let's just say that we have a promoter that comes before our gene. And our RNA polymerase looks for that sequence and uses that to transcribe that sequence into mRNA. It's not a random event. Okay, there's something there. Now, let's talk about how DNA is made. Let's talk about how RNA is made. RNA will be complementary. So RNA will be complementary and anti-parallel, just like a copy of DNA. The only exception is what? The uracil thing, right? Auburn University Gamecocks. AUGC. Now in DNA, the rules are ATGC, but in RNA, it is AUGC. Auburn University Gamecocks. Okay, let's look at an example here. So, number A is a DNA template. We have 5' ACT, CCT, CTT, right? And then 3'. The DNA complement of this is what? 3' TG A G G A G A 5'. ATGC. AT and GC going down the string, right? Okay, that's great. Now, let's look at the RNA complement here. Same DNA, let's keep the same 5' to 3' up here. Now, RNA, 3'. Okay, 3', we're going to keep that same 3'. Now here, remember the rules. Auburn University Gamecocks. AU. So A matches with U, G matches with C, T matches with A. Okay, because right, we have thymine here. We're not switching thymine and uracil, right? We still have the adenine, so we're not going to get rid of that one. All right. C matches with G, G, A, G. T matches with A, and A 5'. Anti-parallel, and everywhere we wanted to have a thymine in the RNA, we have a uracil. Let's give another example. Now, if you've ever seen the movie Gattaca, it was a late 90s movie. Had young Ethan Hawke and Uma Thurman. If you ever saw Kill Bill, you've seen Uma Thurman. Also in Pulp Fiction. Anyhow, if you saw Explorers from 1985, you've seen Ethan Hawke. You've seen him in other things probably. But he's in this movie called Gattaca. G-A-T-T-A-C-A. Kind of like a cautionary tale of a future time when we can sequence people's genomes and what that entails. But the secret of the movie was actually DNA bases. That's kind of the joke of it. Gattaca. All right. Now, what would be the RNA complement of Gattaca? Well, let's go back and put our rules. Auburn University Gamecocks. G, C, A, U, T, A, U, G, U. If we call this 5', we call this 3'. It's anti-parallel, meaning this is 3' and that is 5'. All right? So just basically the rules we've described. Okay, when we transcribe into RNA, we're going to transcribe uracil instead of thymine. But we still do it in an anti-parallel sense. A polymerase still does it, but just remember the uracil change. And again, if you want to remember the base pairing rules here, Auburn University Gamecocks, AUGC, not ATGC like it is in DNA. Okay. Now, this is that promoter idea I was talking about before. Before the start of a gene, we have a site that our RNA polymerase will connect to. And that is a promoter. Okay? There's a lot more to how promoters work than we're going to talk about here. If you are curious about it, please talk to me. I can talk to you about it. But I'm just going to leave it as promoters are what comes before a gene, and it helps to allow the RNA polymerase to connect to it. Okay? And as it connects to it, then it moves down and makes an RNA copy. Now, final thing here is what is a codon. We talked about this before. Okay? A codon is a triplet of bases. And this is how we go from DNA to protein. Now, as I said before, while they were determining this, the central dogma of molecular biology, the huge question was, how do you go from 4 bases to protein and the diversity of life? Well, we don't look at a base per base way. We look at every 3 bases, and every 3 bases is a codon. Okay, so a codon is a sequence of 3 nucleotides in a row, and that codon will code for 1 amino acid. And the order of those codons determines what amino acid is produced. So for example here, let's look at the first one. CCU on the mRNA. Okay, that will make the protein, the amino acid proline. Okay, GAG, glumaric acid, another amino acid. You go down the road, but you see what's going on here. One amino acid does not come from a single base. One amino acid comes from 3 bases. And each 3-base triplet is called a codon. Okay, that's where we get the idea of a codon. That it is not an answer of just one base. How can 4 bases lead to the diversity of life? The answer is it can't. Because it's not, that's the wrong way of saying it, right? We don't go 1 base, 1 amino acid. It doesn't work that way. We look at 3 bases in a row, and that's a codon, and that makes an amino acid. And you do the numbers there, you're going to see that there are 64 possible combinations of nucleotides in a codon, and that is why we get this diversity of life. Okay. Now, we have 64 possible mRNA codons, but I'll tell you that a lot of the codons we reuse. And some of the codons are for what we call stop codons, which will stop protein synthesis. Okay. Now this is a codon table, and you will have to know how to read one of these codon tables for the test. Definitely expect a question like this. Okay? Now, if we were to count every single thing here, right? Each one of these peach boxes has what? Has 4 combinations, right? 4, 8, 12, 16. We keep on counting. 4, 8, 12, 16, so that's what, 32. And you keep on counting, and how many combinations do you see there? You should see 64 combinations. Okay? If you want to count those yourself, you surely can. Now, we have a routine way of converting a codon into an amino acid. So let's say your codon, let's go back to that previous example here. Let's find what CCU codes for. Now here we show it being proline. Let's confirm that on this table. So now we're going to go to the first base. Going from left to right on this table, first base is C. So we see how we have boxes U, C, A, and G? Okay, they're like rectangles. Okay, we're going to go to C. The next one was what? C, C, U. Okay, so C. Then we go over and go to C on the second base, and that's the top row. And then we go to the third base, which is C. U. CCU, PRO, proline. So expect to get a codon on the test, and then take that codon and convert to an amino acid. So for example, I give you a codon AUC. So we go to A, U, C. Okay, we get ILE. We go to, well, let's do another one. Let's do a little bit harder of one. Let's go to A, G, G. Okay, A, G, G, arginine. Okay, as you see, a lot of these amino acids are going to have multiple codons. Another thing you should see is the importance of each base in the codon. Which is the most important of these? The first, second, or third? Well, look at them. It looks like the third one can be variable, can't it? Okay, for example, proline. The third base in that codon can be anything. Okay, histidine and glycine, CAU, CAC, right? You can have some variation in that third base. Okay, we call that the wobble theorem. Now it has to do with how tRNA turns a little bit and doesn't have a full connection with that third base. But for the test, you should know that not every base in a codon is of equal importance. The third base can have some variation in it. So you could have a mutation in the third base in a codon. and you would not be able to perceive it if it changed to the same base that would lead to the same amino acid. Okay? So not every base in a codon is of equal importance. Let's also look at one last thing here, and then we will wrap this up. Let's look at a situation where we have, let's look at the CAU, CAC, CAA, and CAG, histidine and glycine. Okay, where do we see that third base? A and G. What did we call those? Those were the purines, right? Uracil, cytosine, those are the pyrimidines. So when we see this variation, we're going to see that there is a connection with purines and pyrimidines. Okay, so when there is a situation where the third base can be one of two amino acids, we're going to see the tiebreaker here is uracil versus, a pyrimidine versus a purine. All right? We will finish this up with translation next time. We'll talk about how that works. And we'll also come back to this idea of introns, exons, and alternative splicing. We're getting toward the end of class, and I just wanted to start class off with that next time because it's a somewhat difficult concept, and I didn't think I had enough time to give it its full attention. All right, I'll see you Monday.